kick things off, I am happy to invite up Marcelo Bayaderes, Everglades Fellow for the National Parks Conservation Association, to introduce the session and our first speaker. Woo!
Thank you. Hi, everyone. I am Marcelo Valladares. I am the Everglades Fellow with the National Parks Conservation Association, and I am going to say a little thing about NPCA. NPCA has existed for over a century now, which is crazy long. We have over 1.7 million members throughout the nation and over 100,000 specifically in Florida, which is great because there's always a lot going on in Florida. Everglades, Biscayne Bay, very busy. And some specifics, we make sure that all of our national parks funding needs are met, that air and water are clean. Water is very big in Biscayne Bay National Park, as you know. Wildlife is thriving and that their stories are told. On a national level, we are currently focused on investing in our parks' cultural resources, so making sure all the park stories are told, and Biscayne Bay has plenty of stories to tell. So without further ado, I would like to introduce our first speaker, John Halavin.
Good afternoon, everyone. My name is John Halavin. I am a PhD student in the Shark Research and Conservation Lab of the University of Miami under Dr. Catherine McDonald. And I'm excited to share with you today a little bit about my research on great hammerhead resource use patterns in an identified nursery. Before I kick us off, I'd just like to ask that everyone please refrain from taking any images of my data slides as my data are still pending publication. Thank you so much. So a little bit about the great hammerhead. Unfortunately, it is globally assessed as critically endangered by the IUCN. But on a more positive note, here in the Western Atlantic, it is showing signs of recovery. It's likely due in part because of the fact that it is considered to be moderately productive. So then why is it critically endangered? Well, the species is vulnerable for a number of reasons, the first of which is widely regarded as being highly ecologically and behaviorally specialized, which just means that it is highly adapted at using a small subset of resources and behaviors very well. And so a disturbance in those resources could be potentially more detrimental to this species than others. It is still harvested for its fins. It's commonly caught as bycatch in a lot of coastal and nearshore fisheries, and it is highly susceptible to capture stress. And of chief concern to us here is its dependence on nearshore habitats which expose it to a range of anthropogenic threats, including our very own Biscayne Bay, which is the only identified great hammerhead nursery on the east coast of the United States. What's it mean to be a shark nursery? Well, a shark nursery meets 3 criteria. 1, it has a high abundance of juveniles. Those juveniles stay in the area for an extended period of time, and the species uses the area as a nursery repeatedly across multiple years. Great hammerheads were able to satisfy these criteria in Biscayne Bay in 2021. Taking a look at the map on the right, which shows some of our fishing sites that contributed samples to this project, you'll see that site labeled A off the southwest coast of Key Biscayne, where we encounter all of our smallest and young of the year juvenile great hammerheads. And this leads to a lot of remaining questions, such as how do juveniles use the habitat and prey resources that are available to them during their nursery association? In other words, do we potentially see a shift in resource use as they age out of the nursery? When I'm talking about resources, I'm talking about 2 in particular, diet and foraging habitat, diet at a very broad level. Also interested in whether or not we see any sort of shift in juvenile resource use with seasons. To do this, we collected samples from 50 great hammerheads. They ranged in size from about 3 feet to about 12 feet in length. The samples we took were a biopsy of dorsal white muscle and some plasma that we separated from the blood. 37 of the 50 offered us both paired muscle and plasma samples. The remaining 13, we only had muscle for. How are these samples used? These samples were analyzed using a method called stable isotope analysis, which takes advantage of the fact that naturally occurring isotopes in our environment behave predictably and they flow predictably through the food web, such that we can use the isotope carbon-13 as a proxy for foraging habitat, and the isotope isotope nitrogen-15 as a proxy for trophic level, which is In other words, how high you are on the food chain. This is because carbon-13 differs and increases, in fact, as you come in from offshore environments closer to shore into our estuarine and nearshore environments. And our nitrogen-15 increases as you go higher on the food chain. So we can use these two tracers to sort of gauge where an organism is falling in its foraging habitat and on the food chain. The other thing we can take advantage of is the fact that different tissues in the organisms will reach equilibrium with their diet at different rates. The tissues we used in this study were muscle, which equilibrates at around 6 months. It gives us a longer term, stable time average look into the behavior of the individual, as opposed to plasma, which equilibrates at around 2 to 4 weeks, much quicker. Therefore, it's more fluid, more responsive to short-term changes, and gives us a look into real-time behavior. So diving right into the data, first with our muscle nitrogen-15 for that longer term average behavior and against the size of the individual, we're looking to answer the question, does resource use shift with age? What we see is that slight increase in the nitrogen-15 corresponding to a slight increase in the trophic level or food chain position of this individual, of this species as they grow. This is not altogether surprising because we know that our largest great hammerheads are very capable of preying on other large predators, including other sharks. With our carbon-13 tracer, we see a decrease in carbon-13 with increasing size. This corresponds to a shift offshore in larger individuals. Again, not altogether surprising because of the fact that if we remember that map of where our fishing locations were, all of our largest great hammerheads were caught at those offshore sites. If we look at these two isotopes together, we can generate estimates of what is called a niche, or the diversity of resources used by a population or subpopulation. Here we've divided up our 50 great hammerheads into 3 subpopulations. Less than 170 centimeters fork length are our nursery-associated juveniles. We have 170 to 224 as our subadult range, and 225 plus as our adults. The two things we should notice here right off the bat is that our light green ellipse representing our nursery-associated juveniles have the much smallest niche, and relative to the subadults and adults, that is. And we still see a high degree of overlap between all of these niches, indicating that the adults and subadults are still using similar resources as those nursery-associated juveniles. Why is this important? Well, a small niche is associated with specialist behavior using that smaller range of resources. So it seems to be that nursery-associated juveniles are specialists relative to the subadults and adults, which are potentially showing that generalist behavior. And this might mean that our nursery juveniles are in fact more vulnerable to disturbances in whatever critical prey and habitats they are relying on, as opposed to the subadults and adults, which are potentially more resilient and adaptable than we previously thought. Remember, this is a species that is widely regarded as being extreme specialists. Additionally, we see that shift from specialist to generalist behavior. This is actually the opposite of what we normally expect to see in sharks. We expect them to grow in specialization as they grow larger. As they get bigger, they get better at hunting, they get better at finding their optimal prey source, and they get better at overall behaving like sharks. We also see no evidence of niche partitioning or the division of resources amongst groups by foraging habitat and trophic level alone. This might indicate one of two things is occurring in our local Biscayne Bay ecosystem. We either have a high resource abundance, therefore there's low competition, no need to divide up resources amongst each other, or potentially we see competition-driven partitioning that's occurring at a level that we can't necessarily detect using this methodology, such as partitioning diet by specific prey groups or partitioning foraging habitat by season, which leads us to that next question, does juvenile resource use vary seasonally? Up in the top here, we have that our larger individuals. We don't have so many samples of these. We have a lot of our samples on the bottom there from our smaller juvenile individuals, which is what we're more interested in. We've divided up our year into 3 groups. We have our December to March range, which is representative of our dry season down here in South Florida. We have April to May, which is our transition between wet and dry, or dry to wet, I should say, and June to November, which is representative of our wet season range. And what we should notice here in our juveniles in the bottom left is we have a decrease in that carbon-13 signal going into those dry season months, and then an increase in the carbon signal in those wet season months. And this corresponds to a shift to offshore foraging habitats during the December to March range and a shift back inshore during the wet season. Why might we see that? Well, this expansion offshore during the dry season and the contraction back inshore during the wet season might be because of a lower abundance of adults during those months. Our catch data indicates that potentially the adults are less present during those months. This might indicate temporal partitioning or the division of resources such that these individuals are not utilizing the same resources during the same period of time. So what we need to do in the future is to continue to refine the spatial extent of the nursery. We're actively pursuing this using acoustic tagging by putting 10-year acoustic tags in these juveniles. Also, we should look further into determining the extent of dietary specialization. What exactly are the prey groups that these juveniles are feeding on to allow them to be specialists? And we are actively pursuing this using a method called DNA metabarcoding, or getting the DNA from their diet, from their gut. This all has implications for management in that specialization, as mentioned before, necessitates steps to safeguard those critical prey and habitats so that because of their potential greater vulnerability to disturbances, and also the seasonal inclusion of offshore habitats within the nursery boundaries so that these habitats are included in consideration of any management strategies we undertake for the nursery going forward. Thank you very much, and I understand we'll be doing questions at the end.
So we only have 3 talks in this session, and so we have a little bit more time. Does anybody have questions for John at this point before we move to the next talk?
So your smallest shark was 3 feet, you said? They don't start at 3 feet, or do they? So your the hook size could be okay.
For many targeting our larger individuals, those use 16-aught circle hooks as opposed to our scientific long lines, which use either 15 or 13-aught. That's what we use mostly for our inshore fishing.
Thank you.
That's a good question.
Nobody knows. Nobody
The
knows.
same area as we are encountering the primary nursery individuals there off the southern coast of Biscayne Bay. That's where we find those very small human here, recently born individuals. But we're not entirely sure where they are. Yeah, mating, gestation is a fairly long period, so I'm not, we're not exactly sure where the mating grounds are, where these gestating females are coming from, but they are giving birth here. We do know that. They are not directly part of this study, no, but yes, they are going out actively, and we are starting to get preliminary data back from them. The very interesting thing about some of that preliminary data is that it agrees really nicely with what the isotopes are indicating of an offshore shift during those winter months, which is exciting for the prospects of being able to manage those habitats as part of the nursery.
Very nice. If we have more time in between, we can open it up to everybody. But now I would like to welcome up Valentina Katia, Environmental Resources Project Supervisor and Marine Scientist at Miami-Dade County.
Maybe they want to see the screen here.
Every once in a while.
Yes, and where can I go up and
So I'm pretty sure it's forward and back.
Okay.
You can try it out.
forward? Okay, thank you everybody to be here. Could you hear me? Yes. Okay, could you hear me?
Yeah.
I know, but I don't know if, could you hear me? Okay, let me go back. Okay, so today I'm going to talk about the influence of climate change on the physical parameters of Biscayne Bay. This research was done in collaboration with two professors from Rasmas, Josefina Olascuaga and Maria Estebanes, and Elizabeth Mary Jans, who was a graduate student. In this study, we analyzed 20 years of data from 2001 to 2021. We collect the data at 34 stations monthly. So we use surface measurements of salinity, temperature, dissolved oxygen, and pH, the physical parameters. So we evaluate the data, the 20 years data set by decade, season, and zone. So we divided the bay in 8 zones according to the geography, water quality characteristics, and proximity to the coast. So we have 3 zones in the canal mouth zones, the one on the red on the north, that is the North Bay Canal Mouth Zone, Central Bay in dark green, South Bay in brown. And those are the stations or the zones that has more influence of freshwater canals, and they are of course closer to the coast. So then we have North Bay on the extreme north and South Bay here in orange. So those zones are the zones that had restricted circulation and higher residence time. And they are influenced also, the north is influenced by urban, the urban environment. So this area is going to be, we're going to be calling you open bay. So that is the wider area. And so we classify the stations according to the from the coast to the Atlantic Ocean. So the proximity to the coast, in order that they have the similar water quality characteristics. So we have 3 stations closer to the shore. It's called along the shore, the one in dark purple. We have further east the zone called inshore, 3 stations in the middle of the bay. And we have the main bay zone, the one in light green, that are the stations that has higher influence of the water from the Atlantic Ocean waters. So that means that they are more pristine stations. So as a background information, the North Bay is the one that receives more freshwater input. The contribution of 4 canals in North Bay is 57%. The contribution of the Central Bay canals into the bay is 13%. And the freshwater contribution of South Bay canals, so these canals, are 30%. So just for wrap up, so higher freshwater coming out on the North Bay, followed by the South Bay, and the last freshwater contribution is in the Central Bay by these canals. So since we divided the data in decades, so we wanted to know what was the difference between precipitation. So precipitation increased 12%, and the freshwater flows increased 11%. So today I'm going to talk very briefly because it's a very big and comprehensive research, 20 years of data. Okay. So the objectives of this research were to determine if there are changes in the temporal and spatial distribution of salinity, temperature, dissolved oxygen, and pH over a 20-year period. To identify the seasonal and spatial patterns of these parameters and compare them with my previous research, Kacha and Boyer 2005, where we evaluated 10 years of data in the 90s. And to understand the influence of climate change on these physical parameters and the factors that are affecting their distribution, concentration, and temporal variability. Those are freshwater flows from canals, precipitation, water exchange and mixing for the Atlantic Ocean water, sea level rise, etc. So the special distribution of salinity has an increasing gradient from the coast to the open bay, and this has not changed in the last 30 years. So we have been showing here the special distribution of the dry season in March, wet season in September, and November is an example of an anomalous month because it was a very wet year. And even if it's considered the dry season, the canals deliver a lot of fresh water in that. And this can be influenced by the climate change and changes on the seasons. So we use box plots in order to observe the variability between the decades and between the zones and the parameter, right? So what we see here is that 5 zones, including the canal mouth zones, North Bay and South Bay canal mouth zones, they show higher salinity in decade 2. However, while in the open bay, the stations that are closer to the Atlantic Ocean water and has more influence by the Atlantic Ocean water did not change salinity. So the South Bay and North Bay canal mouth zones, there are the ones that has a higher differences between the decades and were 6 units of difference. So in these graphs on the left side, we show an increase, the trends in 2 different areas of the bay. So, North Bay canal mouth zone and South Bay canal mouth zones. And we can see that there is an increasing trend in salinity, and there is a high variability on decade 2. So from 2009 up to 2020. And in order to understand how is the saltwater intrusion in the canals, so we plotted the decadal medians on the right side. We have the decade 1 in blue, the decade 2 in red. So this small icon is the water control structure, and we have the bay to upstream. So stations western or upstream of the water control structure. So we saw that in decade 2 had higher salinity, and that means that is that is an indication of the seawater intrusion. So temperature along this among the zones were higher in decade 2 than decade 1. So temperature is increasing. However, significantly, statistically significantly higher is only the North Bay who show that. Right. And then we can see here that the decadal medians that we plotted. In blue is the decadal median for decade 1. In orange is the decade 2. And we can see that decade 2 had higher temperatures in the bay in all zones except in the South Bay water canal mouth. So the overall temperature difference between the decades was plus 0.5, and North Bay had the highest temperature and the largest decadal difference, 0.8. So that means that North Bay is warming faster. So dissolved oxygen in 4 zones, we saw higher concentrations of dissolved oxygens in decade 2. pH was the only parameter that showed a decreasing trend and lower values in decade 1 in most of the zones. Of course, pH is lower at the canal mouth because of the fresh water, and pH is higher in the open bay in the zones that are closer to the Atlantic Ocean waters. Okay, so as a summary or conclusion, overall salinity, temperature, dissolved oxygen show an increase from decade 1 to decade 2. suggesting that sea level rise and climate change are affecting Biscayne Bay and changing it into a more marine environment. pH was the only parameter that showed a decrease from decade 1 to decade 2, suggesting that ocean acidification is impacting the bay. The spatial distribution of salinity in Biscayne Bay has not changed in the last 30 years, but concentrations have increased over the last decade due to sea level rising. Our results show saltwater intrusion in the bottom layer of some canals. The accelerated phase of global warming has been reflected in a larger and increase in temperature in the oceans and estuaries, and Biscayne Bay followed this trend. North Bay is warming faster than the rest of the zones. Climate change is causing an increase in salinity, temperature in estuaries around the globe, and the alteration of these physical parameters directly affect living organisms, organisms living there. By understanding the distribution, seasonal patterns, and temporal variability of these parameters, we can characterize the health of the bay and improve water management practices in this accelerated phase of climate change. And sorry that I run a lot, but because the setting is kind of difficult, I couldn't show directly with a clicker. Thank you very much. And if there is any question, and sorry. So we acknowledge the people from Miami-Dade County and FIU who has been collecting the data for the last 20 years. Not 20 years, 40 years.
Now we probably have a couple of minutes.
Any questions?
Any questions? All right.
Valentina, concerning your oxygen data, I mean the fish kills in Biscayne Bay have been much in the news, which is due to bacterial consumption of oxygen associated with bacterial blooms. But your data show an increase in oxygen over the period. It's how do you? Thank you.
So the water from the Atlantic Ocean water. The water from the Atlantic Ocean is bringing oxygen to the bay, and we are seeing sea level rising. So in these trends, like the annual means, so we're going to see that. But in fish kill events is something that is just an individual event, a pointed event that is causing, maybe there are a certain costs to that. It's not a general trend. But yeah, and then also, like as I said, it's the Atlantic Ocean water that brings that.
Also, with increasing temperature, you would expect more stratification in the bay waters, which of course would lead to more development of low oxygen in the deeper water too, which is also another, sort of, how do you explain that?
These graphs are surface data. And then in canals is where we are seeing a stratification. That's why we show the stratification in terms of salinity. But unfortunately, I couldn't show all the different graphs in order to, to see the evaluation of other parameters at the bottom layer.
Thank you so much, Dr. Katya. All right, everybody. I think we're gonna do the last presentation, and then we should have a few more minutes if there's any last minute questions here. But round of applause. Very good. Yep. All right. Last but not least, my friend, my steering committee colleague, the incomparable Lauren Janitis, Director of Conservation at Tropical Audubon Society.
Here to talk about birds. Yeah. You guys are the real MVPs for staying to the end. So round of applause for y'all. Great job. All right. So I'm going to put my Tropical Audubon Society cap on. I'm the Senior Conservation Director. And I'm going to be talking about conservation action we've been trying to advocate for the past 6 months now. So my title is called The Birds Are Here, but how do we protect one of the last known nesting rookeries in North Biscayne Bay? First, I'm going to give you a little quick overview on Tropical Audubon Society, though. So we have been around for over 77 years. We actually trace our roots back to the Coconut Grove Audubon Society, which started in about 1915. So we're pretty old. If you want to scan that QR code, it takes you to our website and you can learn more. But we've been advocating for habitats, Biscayne Bay. We helped establish Everglades National Park. We've been around for a very long time. So our mission is to conserve and restore South Florida ecosystems. We focus on birds, other wildlife, and their habitats. And we do that through 3 different pillars. It's education, preservation, and conservation. So with preservation, we have an historic Doc Thomas house. So it was built in 1932. We have a lot of tourists that come visit us. We also do education. So we have our Tropical Audubon Ambassador Program. I'm going to give Dr. Stephanie Clemens, our Education Advocacy Director, a shout out over there, Stephanie. So our adult education program teaching all about different ecosystems here and also how you can advocate for those ecosystems. I encourage you to sign up in August, right? Okay, August time. We also do, we have conservation concerts. We do education events for children. And then for the conservation piece, we do a lot of advocacy work. So that's my role where I'll be defending the UDB, trying to defend wildlife, and speaking at all these different city and county commission meetings. So let's get into the fun stuff. So why we're here today, Mangrove Island Rookery. So essentially, I'm going to give you a little overview here. This is Mangrove Island Rookery. It sits across the Morningside Park. And you guys are probably familiar, it's been in the news a lot lately, is Bird Key up here. So this used to be a nesting rookery, which collapsed in 2019, unfortunately, probably due to human disturbances. And then we have a few different other islands here. This is one of actually the naturally forming islands. These are different spoil islands that were made from dredging. So Mangrove Island sits right here. And what's happening is we're seeing a lot of birds here, and they're nesting here. And so there's some speculation that maybe perhaps since this rookery collapsed up here, they might have moved over here. So FWC actually confirmed that this was a nesting site back in July. We had some advocates from Morningside Park calling us and saying Tropical has to get involved here. This is really important. So what's happening? This entire area is a slow speed zone. So for manatees, do people follow it? They do not. And that's the problem. We're also seeing a lot of folks sometimes fishing is fine, but they fish a little bit too close. And we've seen, unfortunately, birds getting wrapped in that monofilament fishing line and hanging from the trees. So we have seen brown pelicans and a magnificent frigate bird found dead here. So that's what I'm going to talk about today. We also have a photo of that, speeding boats going by that area. Okay, so this is kind of a hard map to see, but this is the FWC slow speed manatee zone. So this is Mangrove Island, this little dot here, and this whole area is supposed to be slow speed. The only time that it changes, this is the Intercoastal Waterway at marker 31, it changes speed from May 1st to November 14th at 30 miles an hour. That's pretty fast. When people are, this is all slow speed, right? And then there are these fast moving boats through here. So if people are kayaking or wanting to visit the other areas, it's kind of dangerous. So we've been trying to advocate too to potentially change that zone all year round to be slow speed. Okay, so we just did a survey. We were out there maybe 2 weeks ago, and we were doing an evening survey, and this is the slow speed sign right here, and you have these jet skis all speeding by. All right, so we started to do these bird surveys, and at first we started to do it in the morning. Birds typically like to, when they're leaving the rookery site, it'll be in the morning time, or at the evening, it's a good time to survey. So we did our first survey in the morning. We did both Mangrove and Legion Picnic Islands. Legion Picnic's a little bit more north in that map that you saw. And you'll see here, we saw brown pelicans and double-crested cormorants and great egrets. They were showing nesting behavior or building nests. Good sign there. We also saw state threatened species. The ones in red are state threatened species, a little blue heron and tricolored heron. And so we broke it up by Mangrove and Legion Picnic Island. Then we decided in March, let's do an evening survey. Let's look at what we're seeing out there. So as you can see, we have fish crows. They loved Legion Picnic Island, but there wasn't a lot of diversity there. We saw so many people, birds at Mangrove Island. And this particular survey was really interesting. So the last one we had about maybe 135 individual birds, 15 different species. This one we had 18 different species, 477 individual birds in March. So we did see little blue herons again and tricolored herons, those state listed species. And the little blue heron was interesting because they were immatures. So that suggests that they were in a surrounding area. They were younger and either they hatched from there. We're not too sure, but it was good to see that Mangrove Island was really being used. And in the most recent survey, so we had a little bit of a reduction here. It was about a little over 250 species. We still had those brown pelicans and double-crested cormorants that were feeding young, doing the whole nesting thing. And we actually omitted Legion Picnic Island. We decided no longer to survey because every time we went out there, Mangrove Island was the spot. We weren't seeing too much going on in the other islands. So we can continue just to document what we're seeing and the behavior there. So this is the fun part. We get to see the pretty pictures. So we have a double-crested cormorant nesting here, and then a brown pelican carrying some nesting material on Mangrove Island. This is my favorite picture. So this is a female magnificent frigate bird. These guys only are documented nesting in the dry tortugas. So we're seeing them now come to Mangrove Island. They're not nesting, but they're showing like nesting behavior. We have females, we have males, and we have immatures that are actually using the island to roost. And then you can see the other bird diversity here. We have a yellow-crowned night heron, great blue heron, and I believe that's a western cattle egret up there. So this is the male magnificent frigate bird. So they blow up their big, Fede, you know the term. Okay. It's like a pouch that they kind of like show it's time to mate and kind of get into that mating behavior. So we had quite a few males on the island. And so then this is a state threatened tricolored heron we saw. And then as I explained earlier with the magnificent frigate birds, this was an immature. So we're seeing different life stages there, right? This is really common too. So what we're seeing is a lot of monofilament fishing line, plastic. There's actually a vessel that's there, but we're trying to encourage people not to go on the islands during high peak time because it disrupts the birds. So it's best to try to leave that, but we are documenting disturbances and I'll explain why. And this just shows you, I mean, this is Mangrove Island. Then you could just see urban Miami in the back and how close this is to the city and how special and we need to protect it. Okay, so how do we do that? That's the question today. So as I said earlier, this is Mangrove Island, Mangrove Picnic Island. And so the slow speed manatee zone is all around this island here. And the city of Miami owns the island. So that's critical. It's really important. Some of the advocacy work we're doing is trying to get the city of Miami commissioners to understand how important it is to protect this island. So how do we do that? So what we're trying to do at Tropical Audubon Society is encourage you all to take action. So we are trying to encourage the city of Miami either to implement a non-motorized vessel zone. So that means no like combustion engines, but you could have maybe a kayaker or paddleboarder go near the island, but basically have a small buffer around it, or designate a critical wildlife area there. So that's something we're really trying to push for. It's been done in other places of spoil islands, and that's either could be a no entry for a certain time of the year or all year round. We also really need increased enforcement. So I don't know if you all saw the enforcement panel earlier with all the different marine officers there, but this is something that's a huge problem here. We don't have enough officers on the water. And part of that is a money issue. So we've been trying to advocate for our state, FWC, the Aquatic Preserve folks, let's get them more money so we can get more marine officers on the ground to write some tickets and enforce that slow speed zone. So not only are we trying to do this for the birds, but the manatees, the seagrass, and for public safety as well. So all of you can do something, and that's why we're really here as Tropical Audubon. How can you help? So unfortunately, I wasn't able to list any upcoming meetings where they have these specifically kind of things on the agenda, but you could always be on the lookout for upcoming City of Miami Commission meetings, Biscayne Bay Watershed Management Advisory Board, or Biscayne Bay Commission meetings. So we have a little group of advocates that have been speaking. I'm really happy because the next Biscayne Bay Watershed Management Advisory Board, they added this to the agenda. So I'm super excited about that. So I hope check their website and I hope to see some of you there. And also the Biscayne Bay Commission meeting, we spoke and asked about slow speed zones, and they took that up at the county level. Commissioner Cabrera and Commissioner Mickey Steinberg really rooted for these slow speed zones. And there's a resolution that the county recently passed to do a study to see different like areas that we might need to implement these slower speed zones, or maybe no boats at all in certain areas. Email or call the City of Miami commissioners. That's the best way to do this. We need a City of Miami commissioner to advocate for this. They own the property. The only way to make that happen is if they back us on this. And finally, sign and share our action alert. So I'm going to put that on the screen. So essentially, if you scan this QR code up here, it's going to have, it's a personalized message. It comes up to our website. And you could either write in your own little letter. It's a generated message that goes to all the City of Miami commissioners. We have it already filled out for you, but you could always edit it and add your little whatever you want to say. And we really need to get them to increase enforcement again. Not only is it an issue for wildlife, but it's an issue for the people there. We're seeing some really interesting things going on where people can even potentially be flipped over on a kayak. We've even seen some of the jet skis chase the birds for sport and try to hit them. So it's kind of an issue there. All right, so I'm going to end there. Keep in touch. These are all the ways you can stay connected with us. And I'm just going to keep that on the screen, though, if you didn't take action yet. All right. Thank you.
All right, we're going to open it up to questions. Any questions for Lauren at this time? Very good.
Thank you for the talk. Thank you for the talk. I have been kayaking to a lot of these islands for many years now. And yeah, I've seen a lot of nests at Mangrove or at, what was the, Mangrove, Mangrove Island. I remember for years at Bird Key, just thousands of birds, multiple different species throughout the years. And I just saw on the news that they're thinking of selling that island because it's privately owned. Is there any kind of efforts that you know of, like of an organization like this to try and keep it in its natural state? Because I'd imagine that those birds will come back to nest at some point.
Great question. So there's an island, just to reiterate, Bird Key. It is up for sale for 31 million. It's privately owned. About 4 acres of it is an actual part of the island, and like 33 of it is submerged land, but that's privately owned. So it's a major concern. So for Tropical Audubon perspective, we are doing interviews. We have met with actually the owners, and we're trying to work to see if we could work together to find a solution where we could like protect that forever. But it's a lot of different, Tropical doesn't have $31 million, so we're trying to see if there's ways we could work with the county, because the mayor actually did make offers back in 2023 through the Environmentally Endangered Lands Program. Unfortunately, the owners did not move forward with that, but we're trying to see if we could work with other groups and maybe we could all come together and buy it or something. We're thinking it through. I do know that there are already bids on it, so we have to act quickly. Yes, but I think if we could all use the power of our voices and say, like, we really need to protect this forever, that's part of what we do at Tropical, is you all have the power to do that as well. So we're happy to work with you, and we can create a little army. All right. Yep. Yep.
I was just wondering if there are official regulations on the islands as it is right now. Like if someone were to get too close, is that illegal? Yeah. No, so it's shouldn't do it, but sorry.
Technically, it's owned by the City of Miami, so it's considered a City of Miami park, and you're actually not supposed to be like fishing super close to shorelines or anchoring at like anything like that, but it's not enforced, and that's because it's really their limited bandwidth. I think when I talked to a City of Miami Marine Patrol, he's like, I have 12 people that has to do the whole area, and they all have to rotate different times. So it's kind of we're trying to find solutions there and try to get more money for them, or where we can increase enforcement. I think there is one more. Okay.
I was actually speaking with a roommate really recently about she like learned that I was, you know, an environmental scientist and in the worlds of ecology. And she was like, oh, well, what do you think are two animals or two species or things that you think suffer the most? And I said, honestly, birds and insects are huge for me. Like, I think that those don't get talked about often enough and are so quickly rejected by society, treated really, really, really poorly, and just like not given the attention that they deserve, equal to like things that are as cool as sharks or dolphins or sea turtles or all these other animals, right? Yeah. But seeing that photo of the islands so that we like got the geographic understanding was such a stark like image for me personally as an ecologist. And all I can think about is the history of the bird species who do nest in those areas, and like whether or not you all have data on how quickly they were displaced from coastal areas prior to development.
So we did do some surveys before my time that we do have data. It's really hard. Like Tropical is a very tiny nonprofit. There's 4 of us. So, but my kind of plans, I would love to form a group where we could start going out more. So we're doing monthly surveys. I would like to do it till January to get a full year. But we do have some historic data. I think the last time, I think it goes to like 2016, but you're right, where that's the Atlantic Flyway, right? So when they used to fly down and stuff, but now with all urbanization and everything, it's like, these are the last spots they got to go to rest. So really important to protect. Did everyone take action? Okay. All right. All right. Thank you so much.